Guys, Miss Cote here, and this is the second video in the heredity unit. In this episode, we're going to work through the stages of meiosis. So let's get started. But before we start, let's just review some vocabulary that I went over in the last video. The concept of homologous chromosomes becomes important in meiosis. Remember that humans have 46 chromosomes organized into 23 pairs. Each pair consists of a chromosome that came from mom and a chromosome that came from dad. These are the homologous chromosomes. In this diagram, we have a cell where the diploid number is 6, so 2n equals 6. The 3 maternal chromosomes are colored in red, while the 3 paternal chromosomes are colored in blue. Notice that these chromosomes have already been copied during the S phase of the cell cycle, so each chromosome is made up of 2 sister chromatids. We can distinguish these chromosomes by their size. So the 2 long chromosomes would be homologous, the 2 medium ones are homologous, and the 2 short ones are homologous. Remember that meiosis is a special type of cell division where we start with one cell and the nucleus will undergo 2 nuclear divisions, so that we'll end up with 4 daughter cells, each having half the number of chromosomes as the cell we started with. Before the process starts, the cell goes through the regular phases of the cell cycle, G1, S, and G2, where everything would be the same as what you learned from mitosis. But because meiosis has 2 nuclear divisions, we separate them into meiosis 1 and meiosis 2. And since each division has the same stages as mitosis, prophase, metaphase, anaphase, and telophase, we use a 1 or 2 after each phase to distinguish between meiosis 1 and meiosis 2. Sorry if this is confusing. Hopefully going through the process will clear things up. Here's a very simplistic diagram of what goes on. Starting with 2 chromosomes, one homologous pair, they will be replicated during interphase, more specifically, the S phase of interphase. During meiosis 1, the homologous chromosomes separate, so at the end of meiosis 1, we have 2 cells. And if you notice, each cell already has half the number of chromosomes. We've gone from starting with 2 chromosomes to only 1 chromosome in each cell. And notice that each chromosome still has its copy. During meiosis 2, the sister chromatids separate, so at the end of meiosis 2, we have 4 daughter cells, each with 1 chromosome. Here we have the diagram from your book, pages 258 to 259, that show more detail. All the phases involved in each division on meiosis. Like I said earlier, the specific phase will either have a 1 or 2 after the name to designate which meiosis it's in, so make sure you remember the numbers when you're discussing the stages of meiosis. Now let's go through the stages. Let's start at prophase 1. Similar to prophase and mitosis, prophase 1 involves the condensing of the chromosomes, the disappearing of the nucleolus and nuclear membrane, the movement of the centrioles, and the formation of the spindle. But a major difference between prophase 1 and prophase in mitosis is a phenomenon called crossing over. Whereas in mitosis, the chromosomes are scattered around randomly, here the homologous chromosomes and their copies come together to form this synaptonemal complex. Looking more closely at what happens during synapsis, here we have the homologous chromosomes. The sister chromatids are being held together by proteins called cohesins. There are also breaks in the DNA sequences at the same places on non-sister chromatids. To bring the homologous chromosomes together, the synaptonemal complex acts as a zipper, joining the maternal and paternal chromosomes. Now that the synapse has formed, the breaks in the DNA are repaired by joining with the other segment of DNA on the non-sister chromatid. This produces the crossovers. Segments of DNA from the non-sister chromatids switch places with the original. The places where crossing over has occurred are referred to as chiasmata. Now that we've covered crossing over, let's get back to the other stages in meiosis 1. Next, we have metaphase 1, where just like in mitosis, the chromosomes line up at the metaphase plate. However, different from mitosis, it's the complex of homologous chromosomes that line up at the metaphase plate. This is where independent assortment takes place. Notice in the diagram that when the complexes line up, there are 2 reds and 1 blue on the top. Independent assortment is the randomness of how these chromosomes orient themselves, and eventually how they're sorted into the daughter cells. We'll discuss this a little bit more later on. The homologous chromosomes separate during anaphase 1, each chromosome and its copy moving towards the opposite sides of the cell. And when they each get to their poles, telophase begins. Depending on the species, sometimes the chromosomes uncoil and the nuclear envelope comes back and all that stuff, but either way, cytokinesis will split the elongated cell into 2 distinct cells. Remember, if it's an animal cell, then a cleavage furrow will form. If it's a plant cell, the cell plate will form and eventually elongate into a new cell wall. At the end, 2 haploid daughter cells are formed. At this point, the cells are ready to undergo meiosis too. Since each cell already has chromosomes made up of 2 chromatids, the daughter cells do not need to run through the cell cycle and replicate the DNA again. It's already done. So let's move on to meiosis 2. Looking at the pictures in the diagram, meiosis 2 is very similar to mitosis. If the chromosomes uncoiled and the nuclear envelope came back during telophase 1, then the chromosomes will recondense and the nuclear envelope will disappear now in prophase 2. The centrioles start moving and the spindle forms. The chromosomes line up during metaphase 2. The sister chromatids separate during anaphase 2. And telophase 2 has the chromosomes uncoiling, nucleolus and nuclear membrane coming back, and the spindle disappearing. After the cells physically separate during cytokinesis 2, this produces 4 daughter cells, each with half the number of chromosomes. And now that you're familiar with how both mitosis and meiosis work, it's always a good idea to be able to compare the two, especially when it comes to the differences between them. So here on the left is mitosis and on the right is meiosis. Starting with the parent cell where 2n equals 6, so there are 6 chromosomes in the diploid cell, mitosis will conserve the number of chromosomes. Each of the 2 daughter cells have 6 chromosomes. And look, they're an exact copy of the original chromosomes. On the right with meiosis, we end up with 4 daughter cells, and each only has 3 chromosomes. So we've reduced the number of chromosomes by half. And looking at those chromosomes, all 4 of those cells are genetically different from each other and obviously from the original parent cell. And here's another nice visual from your book reviewing the properties of both mitosis and meiosis. I'm not going to go over all of this, so you can always pause the video if you want to copy this down, or you can find it in your book. In addition to the number of divisions, the number of daughter cells produced and the amount of chromosomes in those daughter cells that we were just talking about, there are 3 very important events that only happen in meiosis. And this stuff only happens in meiosis 1, actually. First, crossing over happens during prophase 1. Second, the chromosomes line up as homologous pairs instead of individual chromosomes during metaphase 1. And third, at anaphase 1, we see the separation of the homologous chromosomes as opposed to separating sister chromatids. And why is all this important? Because remember that meiosis produces the gametes, eggs or sperm, for sexual reproduction. And the goal of sexual reproduction is to produce offspring that are a unique mixture of the 2 parents, which contributes to genetic variation within a species. And while mutations do happen and are the original source of genetic variation, most genetic variation comes from meiosis and fertilization. More specifically, crossing over, independent assortment of the chromosomes, and random fertilization. I know we've already talked about crossing over when we went over prophase 1. Segments of DNA are exchanged between non-sister chromatids. This produces what we refer to as a recombinant chromosome. Recombinant meaning that the chromosome is a mixture of DNA from different sources, mom and dad. Since the chromosome is no longer just mom or just dad, it's a mixture of both. This contributes to genetic variation. See how all 4 daughter cells are genetically different from each other. To explain a little more of what happens during metaphase 1, let's look at this diagram. I said earlier that when the homologous chromosomes line up, how they line up is completely random. Here on the left, the blues are lining up on the left side of the equator and the reds are lining up on the right side of the equator. So at the end of meiosis, the two combinations are that two cells are all blue and two cells are all red. On the right, the bottom chromosome is flipped and now blue is on the right side of the equator. So at the end of meiosis in this configuration, the daughter cells have a combination of a red and a blue chromosome. This is a representation containing only two homologous pairs of chromosomes. The more chromosomes in a species, the more possible combinations that we can have. The rule of thumb is to calculate the number of possible combinations in a gamete, it would be 2 to the n, where n is the haploid number of chromosomes. So in the case of humans, where our haploid number is 23, 2 to the 23rd would give you more than 8 million possible combinations in an egg or a sperm. And that's a lot of variation. Last is random fertilization. This is simply which sperm is going to fertilize which egg. So in humans, if an egg with more than 8 million possible combinations fuses with a sperm also with more than 8 million possible combinations, then the zygote, that very first cell, would be the product of the two possible combinations, which would be over 70 trillion combinations. This makes each person unique in their DNA fingerprint. And that's pretty much it for this chapter. Hopefully this clears up the process of meiosis and how mixing up the chromosomes contributes to genetic variation, producing offspring that are a mixture of the parents. So until the next time I see you, be a good human.